study the microbiome. My name is Bhatt. I'm an assistant professor of medicine and genetics here at Stanford. I've only been here just under 2 years, and I'm the most recent awardee. So I think technically the award starts on September 1st, so I'm like T-minus 10 days. So I'm going to tell you a little bit about the project that I was a little eager, so I got started anyhow. I'm going to tell you a little bit about the project that my laboratory is going to be studying. So I'm a physician scientist. I was trained in hematology and oncology, and now I'm a geneticist, though I used to be a biochemist. I guess I'm lapsed now. And the title of my project is The Microbiome, Extremes of Body Mass Index in the African Continent. So I studied the microbiome, and I think they chose to have me speak last so that lunch would be out of the way. It's okay, I use a lot of euphemisms, so it's perfectly fine. We live in complex symbiosis with microorganisms. Lots of them, like 100 trillion microbes. These are bacteria, viruses, and fungi that live within and on every single one of us. They make up about 3% of our human body mass, so getting rid of them would be a really great way to go on a quick diet. There's a raging debate on whether there are as many microbial cells and particles as there are human cells in the body, or maybe even 10 times more. And there are 10 to 100 times as many microbial genes as there are human genes. So our genetic variation comes in large part from our genome, but potentially even in larger part from our metagenome. We call these genes that are present within microbes our metagenome. As you all probably know from the popular press or from your scientific reading, the largest number of microbes that we have are within our GI tract. And when we think about where we acquire microbes from, microbes are actually the perfect place to investigate gene-environment interactions. Okay? We just fed ourselves, we also fed our microbes, and so our microbes selectively grow and die based on what we eat. Our microbes are also dramatically shaped by our own genetics. So we first learned that we had a microbiome a long time ago. This seems like a new kid on the block. It's really not. In 1677, about 300 years before I was born, Anton van Leeuwenhoek, who was credited with discovering or inventing rather the light microscope, did the experiment that I think any one of us would probably do, which is he said, I'm going to look at some stuff under the microscope, and he took a toothpick and he scraped his teeth and he put it on a glass slide and he looked at it. And he drew these amazing pictures. This is what he found in the junk between his teeth. This was his plaque, and they didn't have great dental hygiene then, but even now if we did this, what we would find is these tiny little organisms. Some moved, some didn't, some were round, some were long, rod-shaped, some were kind of spiral. And what he discovered actually was teeming in his mouth was bacteria. And these were bacteria, although we didn't have a word for bacteria then. He decided that they were like small animals, and so he called them animalcules. And this was actually the first description of these microbes that live within us, obligatorily. So I'm going to summarize the science that my lab does in one slide, which is we take samples from humans, we sequence them, and then we categorize all the sequences that we find. There are, in every single one of us in this room, bacteria and viruses that have never yet been discovered or described in the literature. So each one of you is like a discovery waiting to happen. And that actually poses a lot of interesting genetic and computational problems because it's easy for me to find these organisms that have been described before because I can look at the sequence and I can match it up to a reference and say, yes, this looks like the blue bacteria, give it a name. But many times I'm left with sequences that I can't identify at all. So we spend a lot of time thinking about how to deal with this information. Just a quick overview of what happens in the has happened in the literature over the course of the last 15 years or so. We've learned that babies actually first get their microbes from mom, and they actually have differing microbiomes based on whether they are born by C-section or by a vaginal birth. And so what you can see over here is the pink is a vaginal birth, and the red dots are the baby's gut microbiome from babies born by vaginal birth, very similar to one another. Over in the light blue and the dark blue dots are the skin microbiome of the mom, and the light blue dots are the gut microbiome of babies born by cesarean section, right? Because when you're born by cesarean section, baby is first exposed to the mom's skin. So we can do this stuff. This is a another beautiful paper came out of Jeff Gordon's group where they looked at the number of different types of bacteria that they could name over time. And this is age on the x-axis. And you can see that we start with a very small number of microorganisms. Number of microorganisms is on the y-axis, and it blossoms over time. And that happens whether you're in the US in blue dots, a Malawian in red dots, or Amerindians in green dots. We know that very early on is where most of the change is happening, and that's because we think both the baby's immune system is developing, which controls some of the microbes that are present, but also they're introducing lots of new foods and exposures. We know that gut microbes can induce obesity in animal models. It was beautifully shown that if you take the microorganisms from this fat mouse, this is a genetically fat mouse, and you take those gut microbes and you put them into a skinny mouse, you can make the skinny mouse fat. We also know from the literature that certain microbes are associated with protection from stunting and malnutrition. Right? So not only can microbes make you fat, but maybe microbes can keep you healthy if you are predisposed to stunting. And what we know more than anything else is that we have a major problem in this world with cardiometabolic disorders and malnutrition. We classically think of malnutrition as skinny kids, stunted kids, so short kids. But now we've actually started to identify this as the double burden of malnutrition, which is defining both stunting and wasting, as well as obesity, as two different ends of the same spectrum here. They are both forms of malnutrition. Nowhere is this more prevalent than in Sub-Saharan Africa, where we're seeing rapid industrialization, which means that you have both of the problems of the ends of malnutrition. This is information from a WHO report. You can see up top the percentage of children under 5 affected with stunting or wasting adds up to 34%. The percentage of children who are overweight is 14%. If you take a look at the prevalence of adult overweight and obesity, you can see that both sexes actually have a high rate of, in blue, overweight and in yellow, obesity. And this problem is even worse in women than it is in men. And so understanding that the microbiome impacts how we consume our nutrients and can impact both ends of the spectrum of malnutrition, both wasting and obesity, we decided to study this in the lab. The microbiome is a hot topic, which is a good thing as an assistant professor, because in order for me to stay on and get promoted and get grants, I need to work on a hot topic. I happen to also find it very interesting, but this is on the x-axis the year and on the y-axis the number of publications in the field, and you can see the microbiome is booming through the roof. But that actually is the number of publications that involve Sub-Saharan Africa. So Sub-Saharan Africa has about 1/7 of the world's population, and the number of papers, I've actually read them all. Like, that should tell you something. Actually, I had a medical student this summer do a systematic review to identify all of these papers. He's also read them all in a summer. There's about, between 40 and 80, depending on how you count them, primary work. So again, the problem that Andres told you about. And so what we want to do is identify microbial biomarkers that are associated with the extremes of BMI in the African continent. In order to do this, we're collaborating with some very talented researchers and in many countries within Africa to overlay our microbiome research, which is a nice way of saying we're collecting poop, on their beautiful genomics research that is seeking to identify population structure variation and alterations in the human genome that associate with cardiometabolic disorders. So this is actually where I went. I'm just going to briefly, or in like 2 minutes, tell you about my first efforts to try to get this project going. I started in Johannesburg, which is right over here, and that's where one of our research sites is. Another collaborating research site is right near Kruger National Park, which you can see is labeled with a blue square, right near the Mozambique border. In relief, this is actually what the area in South Africa looks like. There are 2 yellow stars in the kind of upper right quadrant. And the middle star is Johannesburg, which actually now you can see based on that relief is in a high plain. And the other location is actually down beyond the mountains in a low plain near Kruger National Park. So they're about 6,000 feet different in elevation and about as different as places can be in terms of how people live. So the location that we're studying within Johannesburg is a traditionally black township called Soweto. There in Soweto, this is kind of what it looks like. miles and miles of shanty towns and even houses. This is where a predominant a number of black Africans live in Johannesburg. You probably recognize these famous Orlando Towers. These are right in the middle of Soweto. Lifestyle in Soweto, it's crowded, it's kind of dirty, people live very urban lives, they get access to fast food, they experience a lot of malnourishment, but malnourishment typically at the high end of that spectrum with a very high incidence of obesity. The other location, as I mentioned, is you drive 6 hours east, and there's no better way to get to know your collaborator than to do a 6-hour road trip with them, I assure you. And you end up going down through this mountain pass to the lowlands. And what folks there have done is for the last 25 years, they've been doing health and demographic surveillance in this area of very closely connected villages. That means that they have followed every single man, woman, or child, birth, death, marriages, in-migration, out-migration, health status of now over 100,000 people. Every single one, in every single house. This was amazing to me. This is kind of what it looks like. So this was a place that we visited. This is another example of what this region looks like. And to touch back to the science for a moment, what we plan to do with the prize money is to start a pilot study and then a future study. The pilot study will take 100 postmenopausal women in each of the 2 study sites, the urban site, Soweto, as well as the rural site, Agincourt. All of these women happen to be enrolled in this larger AWIGEN study, so we're collecting a lot of very exciting demographic information and anthropometric data already. We have a lot of clinical data that have been collected, and what my laboratory is going to do is to provide the microbiome aspect of this study. After the completion of this pilot protocol, we're actually hoping to extend this to sites in 3 other countries in both Eastern and Western Africa. This is actually the study site headquarters, so this is where I go and stay. It is an open area right near the Kruger National Park. Apparently a leopard lives on the premises. I did not see it, but I did find its footprint. I described to you that these folks do health and demographic surveillance. When they started 25 years ago, this is actually what they took door to door. It was this little piece of paper. You probably can't read it, but it has things like, what's your village number, what's your house number, your first name, your surname, your sex, your age, a few more columns. This is now done in a much more high-tech way. These are members of our collaborating team. This is a gentleman who's actually showing me, this is Bernard, he's showing me the maps. So this is actually a map of the entire village. And each of those village houses has a number on it, and that's how we keep track of all of the individuals within these houses. This is the woman who's actually in charge of all of the data, so all of the historical data is still kept in paper, but backed up to the cloud, of course. And so she's standing in front of the data that they've collected. And these are the 2 main collaborators of mine. To my left is Scott Hazelhurst, who's a bioinformatician. He's actually trained in computer science, but like me, has found a passion in the microbiome. And to my right is Stephen Tolman. Stephen Tolman, he and his wife, physician scientists who had this crazy idea 25 years ago to take a car out there and start to count people. They have incredible lab facilities. So this is actually what the lab facility looks like in the middle of this rural area. This is Imitrude, who is in charge of the lab. She's very proud of it. They have a much nicer label printer than we do in my lab. My lab pointed that out to me. They have -80 freezers, they have centrifuges, and they have everything that you need to be able to do lab processing, which is very cool. But when you go to get samples, we don't wait for people to come to us. They have to travel many, many miles, tens and tens of miles to come to us, so we go to them. And so we actually went out to some patients to collect, in this case, nasopharyngeal swabs. In order to make sure that the swabs are actually kept at the right temperature, you put them in a styrofoam box with an ice block. That little thing with the wire is actually a temperature monitor. And up on the top, you can see they actually monitor the temperature at which they keep their samples. I was so impressed by this. This is actually the team, the nurse, and now the field worker who are setting up their little table, which they carry with them underneath a marula tree. So every house has a little marula tree to give them shade. And this is where we were collecting samples. And this is our poor little victim who ended up having to get a little nasopharyngeal swab taken because they're looking at respiratory virus transmission among individuals. But we decided to kind of go along with this effort to try to understand how we might collect samples. And it was amazing because we were able to talk to villagers, participants, field workers, nurses, to figure out how we were actually going to collect the samples we needed for this study. So with that, we are actively collecting samples for the study. We hope to have our first 200 by the end of this year, and hopefully next time I'll have some data to show you. So with that, I wanted to thank the Center for Innovation in Global Health, which actually gave me a small seed grant to help support this travel and really push this research forward. And a very big thanks to the Rosenkranz family and friends for making this possible for me.
Well, thanks very much. That was extraordinary. I just want to take this, I'm going to turn it over to Ricky for the last remarks, but I just want to take this opportunity to say thank you again, congratulations, happy birthday. I want to thank the Rosenkranz family. Ricky and Bobby have worked with us tirelessly, all your friends and colleagues who have helped support this. I hope you see the extraordinary things that this is enabling, which is one of the most satisfying things for us, is to see these young scientists being able to pursue the work that is really going to make a difference for people. So with that, thanks again and happy birthday.
Well, thank you everybody for joining us today. Doug, what a wonderful event. I can't think of a better way to celebrate your birthday than listening to the bright future of science, because that's really what your mission has been. So even though we wanted to do this and we didn't know exactly how it was going to turn out and when and where, it is amazing that August 20th we spent the better part of the afternoon listening to the bright future that lays ahead of us. And this bright future is here because of the foundation that you have left for all of us. Eran, Andres, Sanjay, Ami, and of course our other two winners, we are so excited what you're doing. We can tell that the prize played a role in your life, is playing a role in your life. We want to ask you not to forget the main thing that my father has taught all of us, which is that it is incredibly important to become a mentor. So now it's your turn to also continue and mentor all the other people that are around you. And we can't wait to hear where your lives go, and we can't wait to hear what the next dozens, hopefully, of Rosenkranz Prize winners, scholars, come and tell us. Thank you to FSI. Thank you, Doug. Thank you to Stanford. And let's continue celebrating our father. Well, and I'm just going to say, because Bobby started this thing last night, and I think we need to keep with it, which is great. There is a saying in Yiddish that, when somebody has a birthday, you want to tell them to continue on their life. And in Yiddish, it's not 100. 100 isn't the end of the road. It's 120. So Bobby taught all of us in the room yesterday how to say it. It's 2 words, bis 120, technically 3, but it's really 2. So can everybody in the room join me, join Bobby, join the family in saying to my father, bis 120. Thank you. And Stefano, sorry. Thank you so much for such. Thank you for such an inspiring way to begin this, because you show us the way things really ought to be done, and your success is beyond measure. Thank you.